All right, let's dive right in. Today, we're going to peel back the layers of biology and look at the beautiful fundamental chemistry that makes all of life possible. And you know what? It all starts with just a few simple molecules and some really, really clever rules. So here's our game plan. We're going to start with the stage where all the action happens, water. Then, we'll get into the delicate balancing act of pH and the systems that protect it. From there, we'll meet the stars of our show, simple sugars, and see how linking them together creates the difference between, say, food and fiber. And finally, we'll wrap it all up to see why this chemistry is so important. Okay, first up, the single most important molecule for life as we know it, water. I know, it's so common we barely even think about it, but its unique properties are literally what make biology work. I mean, think about it. The majority of your body is just water. It's not just filling up space, though. It's an active, absolutely essential participant in everything. And I mean everything that happens inside your cells. So what's its secret? Well, it all comes down to one simple fact: water is polar. The oxygen atom kind of hogs the electrons, which gives it a slight negative charge, leaving the hydrogens slightly positive. Because of this, water molecules love to stick to each other through these things called hydrogen bonds. This also makes water a fantastic solvent for anything else with a charge. And at the same time, it shoves nonpolar things, like fats, together into clumps. This is what creates compartments and helps proteins fold. It basically builds the entire structure of the cell. It's the matrix of life. But water has this secret personality. See, it isn't just the stage for the play. Sometimes it's one of the actors. A tiny, tiny fraction of water molecules do something really interesting. They split apart. Yup, a water molecule can break into a hydrogen ion, that's an H+, and a hydroxide ion, an OH-. And the concentration of those free-floating hydrogen ions is what we measure with the pH scale. You can think of it as the master dial that controls the entire chemical environment inside our bodies. The best way to think about it is like a seesaw. At a neutral pH of 7, like in pure water, the H+ and OH- ions are perfectly balanced. But if you add more H+, the solution becomes acidic, and the seesaw tips down to a lower pH. If you add more OH-, which mops up the H+, the solution becomes basic, and the seesaw tips up to a higher pH. And get this, the scale is logarithmic, which means a pH of 3 is actually 10 times more acidic than a pH of 4. And this just shows how wild the range of pHs is in our world. Your stomach acid, incredibly acidic, around a pH of 2. A can of cola, not that far behind it. But your blood, your blood is kept in this super tight, slightly basic range, right around 7.4. To put that in perspective, that cola has about 10,000 times the acid concentration of your blood. So that brings up a really important question, right? If your blood is so sensitive, how can you drink a can of cola without, well, causing a major problem? How does life maintain that perfect balance? The answer is one word: buffers. These are the unsung heroes of our internal chemistry. They're the guardians of our pH stability. So how do these little shock absorbers actually work? Well, you can think of a buffer as a kind of chemical sponge. It's made of a pair of molecules that can either soak up any excess acid, or they can release a little bit of acid to neutralize a base. It's this pair, this weak acid and its partner, that works tirelessly to keep the pH stable. And the effect is, honestly, it's staggering. Let's just imagine we add the same amount of a strong acid to two different beakers. One's got pure water, and the other has a buffered solution. What do you think happens to the pH? In the pure water, the pH just tanks. It absolutely plummets. The system has zero defense, and it becomes extremely acidic almost instantly. But in the buffered solution, look at that. The pH barely even budges. The buffer just absorbs the shock, keeping the whole environment stable. This is exactly what the bicarbonate buffer system does in your blood every second of every day, keeping your pH locked in at 7.4. It's absolutely essential for survival. Okay, so we've set the stage with water and pH. Now let's bring on the main characters that actually live and work in this environment, the carbohydrates. We'll start with the simplest of them all, sugars. So these monosaccharides are the basic building blocks. Chemically, they're defined by their specific arrangement of carbon, hydrogen, and oxygen. The most famous one, of course, is D-glucose, the #1 fuel for our cells. And here's a cool thing. While we often draw them as straight little lines, In the watery world of a cell, they actually fold up on themselves to form more stable ring shapes. And this is where the story gets really, really cool. By linking these simple sugar rings together, nature creates these massive molecules with wildly different properties, and the whole thing hinges on one tiny, almost invisible detail. Just think about this for a second. What's the real difference between a potato, which is mostly starch, and a tree trunk, which is mostly cellulose? I mean, they're both made from the exact same building block, glucose. So what makes one soft and digestible and the other hard and fibrous? The answer is just, it's so simple, it's brilliant. It's the bond. In starch, the glucose units are joined by what's called an alpha linkage. This bond puts a little kink in the chain, which makes the whole thing coil up into a loose helix, perfect for storing away energy. But in cellulose, the units are joined by a beta linkage, and this tiny change in the geometry makes the chain perfectly straight. These straight chains can then line up next to each other and form hydrogen bonds, creating these incredibly strong, rigid fibers. And that's the punchline right there. A simple flip in the orientation of a single chemical bond is what turns a glucose polymer from digestible food into one of the strongest structural materials on the planet. Your body has the tools, the enzymes, to break alpha bonds, but not beta bonds, which is why you can eat a potato, but you can't eat a tree. And nature uses this trick all over the place. Glycogen, which is how our bodies store energy in our liver and muscles, is basically just a super branched version of starch. Chitin, the tough stuff that makes up insect exoskeletons and the cell walls of fungi, is another polymer of modified glucose, linked up just like cellulose to be strong and protective. It's all based on the same simple idea of linking sugars together in different ways. So let's zoom out for a second and put all these pieces together. Why does all this fundamental chemistry actually matter? Because it shows us something really profound about biology, the incredible, mind-boggling complexity we see in living things, from how our blood stays perfectly balanced to the structure of an entire forest. It isn't based on infinitely complex rules. No, it emerges from a handful of simple, elegant chemical principles that are just repeated and combined in the most ingenious ways. And that leaves us with a pretty cool thought to chew on. If the difference between food and wood really just comes down to the direction of a single bond, what other seemingly small chemical details are out there, quietly orchestrating the complex biological world all around us? It's a fascinating question, isn't it?